Hi everybody, welcome back. I'm going to try to go through this. I seem to be losing my voice, but I think we can manage at least this section. Today we're going to be covering glycolysis, specifically the steps in glycolysis, so the actual pathway, as well as regulation. So the specific sections that we're covering from your chapter in this lecture is going to be sections 1, 2, and then the one on regulation, which is, where are you? Sorry, I'm flipping to the textbook now. So I think I changed it. So specifically in this lecture, I'm going over sections 1, 2, sections 4, and I don't get into sections 5 yet. Okay, so just bear with me. We will get to section 5 in another lecture. Alrighty, so here are your learning objectives. You'll see specifically there's not as many as in other chapters because we're kind of breaking this chapter up into the various different sections, and this way we can cover more of the concepts in detail instead of glossing over a number of different concepts. Sorry, I had to get something to drink. Okay. So first off, glucose, which is our starting material of glycolysis. As we know, glucose is the major source of metabolic energy in most cells. It usually appears in the blood from the breakdown of polysaccharides, such as glycogen or starch, and then enters into the cell, being transported by a specific carrier protein. Glucose is brought into the cytosol of the cell, which is where glycolysis occurs. Sorry, our little glucose molecule on this picture is not very clear for some reason, but here is our friend glucose. Okay? So once glucose is brought into the cell, we undergo glycolysis. So here's our overview, or our big picture of glycolysis. So our summary of the reaction is that you have glucose plus 2 NAD plus 2 ATP plus 2 inorganic phosphates yields 2 pyruvate plus 2 NADH, 2 ATP, and 2 water molecules plus 4 protons. So as you can see, from one glucose, we're able to yield a net of 2 ATP molecules, 2 NADH molecules, 2 pyruvate molecules, 2 molecules of water, and 4 protons, or 4 acidic protons. In glycolysis, glucose is converted into 2 C3 pyruvate units, which you can see here. So C3 meaning it's got 3 carbons, which generates enough free energy to synthesize a net gain of 2 ATP. So we see in our first stage, which is this stage here of glycolysis, we actually burn 2 molecules of ATP. However, in our second stage, we make a total of 4 molecules of ATP, giving us a net gain of 2 overall. And if you look, glycolysis can be broken down into 10 basic steps. So we got 1, 2, 3, 4, 5, 6, 7, 8, 9, and there's a 10th one hidden somewhere that I seem to have skipped, but we will see it when we go step by step. Okay. So as I sort of touched on a second ago, glycolysis can be divided into 2 stages. So stage 1 is where energy is invested. So we consume 2 molecules of ATP here. So energy is invested to phosphorylate and cleave glucose into 2 molecules of the triose glycerolaldehyde 3-phosphate, or we call this G3P for short. So that consumes 2 molecules of ATP. In stage 2, energy is recovered with the 2 molecules of glyceraldehyde 3-phosphate are converted to pyruvate, generating 4 ATP molecules. So you can see we generate 1 NADH and then 4 molecules of ATP. So I'm sorry, it's 2 NADH and 4 molecules of ATP, giving us, again, the net gain of 2 ATP per glucose in glycolysis. Now, at the end of glycolysis, as you can see, we also end up with 2 molecules of pyruvate. So pyruvate is then further oxidized in either aerobic respiration, so it gets fully oxidized into CO2, H2O, and protons, releasing energy to do oxidative phosphorylation to form ATP, as well as some more NADH is formed. Or we can undergo anaerobic oxidation. You can do alcoholic fermentation with bacteria, is where you get ethanol and CO2. Or in human, or in our cells, or our muscle cells, we can undergo homolactic fermentation, where we get lactate buildup, as well as regenerating our NAD+ to use back in glycolysis. We'll touch back on that later. So let's go through glycolysis step by step. So in step 1 of glycolysis, we are synthesizing glucose 6-phosphate from glucose. So we have glucose plus ATP, and we transfer the phosphate from ATP over to glucose 6-phosphate in order to form glucose 6-phosphate, ADP, and a proton. Now, this reaction is governed by hexokinase, and hexokinase couples the ATP dephosphorylation with the phosphorylation of glucose 6-phosphate in order to make the reaction go in the forward direction. So let's just read over this real quickly. Immediately upon entry of the cell, glucose is phosphorylated. Phosphorylation of glucose prevents the transportation of glucose back out of the cell. So once we phosphorylate glucose, we can no longer go back into the bloodstream, into the cell, or out of the cell. This reaction is catalyzed by hexokinase, which is a transferase enzyme. It consumes 1 molecule of ATP, and then if you notice, hexokinase also requires magnesium as a co-substrate. This reaction is irreversible, basically irreversible. There is some cases where we can reverse this reaction, however, it is very rare. And we'll see that in gluconeogenesis, what needs to occur for this to happen. So, however, just for all purposes for glycolysis, this reaction is irreversible. Glucose 6-phosphate itself, so the product of this reaction, is actually an inhibitor, an allosteric inhibitor of hexokinase. So it acts as a feedback inhibitor in this pathway. So if we get an accumulation of glucose 6-phosphate, it will bind to hexokinase and inhibit further phosphorylation of glucose. This way, if glucose builds up in the cell, it can then enter back into the blood for other cells to use. Phosphorylation of glucose to glucose 6-phosphate requires approximately 15 kilojoules per mole. Breaking the phosphoanhydride bond of ATP, however, releases 30 kilojoules per mole. So if we have 15, we require 15 kilojoules per mole, but the breaking the phosphoanhydride bond releases 30 kilojoules per mole. That gives us a net of negative 15 kilojoules per mole for the overall reaction, making it overall spontaneous and thermodynamically favorable. So why is magnesium needed by hexokinase? So the reason magnesium is used by hexokinase as a co-substrate is that it will shield the negative charges of the alpha and beta phosphate groups, making the gamma phosphate more accessible. Making the gamma phosphate more accessible for nucleophilic attack during glycolysis. So the oxygen on carbon 6 can come in, nucleophilically attack that phosphate group, and that negative charge isn't so electrostatically repulsive that the glucose can get in close enough to do that. Alright, so step 2. In step 2, we're converting glucose 6-phosphate to fructose 6-phosphate. This is done using an isomerase, PGI. What we're doing is we're converting an aldose of glucose 6-phosphate to a ketose. This is a readily reversible reaction, and what happens is that this makes the C1 of the fructose product, this guy right here, available for phosphorylation in our next step. The proposed reaction mechanism for this involves a general acid-base catalysis with an enediol intermediate. You can read up more about this specifically in your textbook. However, this is about as detailed as I wanted to go in on this lecture. We can talk about this more in class and as we go over the POGL activity. In step 3, we now have phosphorylation of fructose 6-phosphate in order to make fructose 1,6-bisphosphate. Again, this is using another kinase, phosphofructose kinase, requires magnesium as well as another molecule of ATP to transfer that phosphate group and to couple the energy of that reaction. So here again, fructose 6-phosphate is phosphorylized at C1 to make the fructose 1,6-bisphosphate. This reaction is catalyzed by a transferase enzyme, phosphofructokinase 1, requires magnesium, and another molecule of ATP. This reaction is made spontaneous, again, by breaking the phosphoanhydride bond, and is therefore reversible. At this point, the cell is committed to glycolysis. Now, phosphofructokinase, or PFK1, is allosterically inhibited by high levels of ATP and citrate. So ATP and citrate are both byproducts of pyruvate oxidation. So if we have enough ATP and we generate a lot of citrate, that indicates that the cell has enough energy that it needs, and it doesn't need to break down any more glucose. So once we turn off phosphofructokinase A by inhibiting it, we'll get a buildup of the fructose 6-phosphate, which will then convert back to glucose 6-phosphate. We'll get a buildup of glucose 6-phosphate, which will then inhibit hexokinase, and then once we inhibit hexokinase, the glucose that builds up in the cell will then be transported back to the blood. All right. However, we can also activate phosphofructokinase by AMP. So if we have a high level of AMP, it will bind to phosphofructokinase 1 and increase its activity so that we create more of this committed product, the fructose 1,6-bisphosphate, which will then cause more glucose to be consumed. Step 4. We have cleavage of FBP, or the fructose bisphosphate, is cleaved by an enzyme aldolase into dihydroxyacetone phosphate, which is right here, and glycerolaldehyde 3-phosphate, which is right here. This reaction is an aldol cleavage, leaving us an aldehyde and a ketone as the products of the reaction. This reaction is actually thermodynamically unfavorable. We get about a positive delta G. However, it occurs because the products, glycerolaldehyde 3-phosphate, as well as DHAP, are rapidly removed. So this is controlled more by equilibrium than by thermodynamics in this case. Alrighty. And again, this can be reversed. All right, so in step 5, the dihydroxyacetone phosphate, or DHAP, is isomerized into GAP, or the glycerolaldehyde 3-phosphate, because glycerolaldehyde 3-phosphate is what is continued to be used in glycolysis, not the dihydroxyacetone phosphate. So these can freely isomerize back and forth through an enediol intermediate, which is catalyzed by the enzyme triose phosphate isomerase. But because GAP proceeds rapidly through glycolysis, this reaction is favored, the DHAP conversion to GAP, because the concentration of GAP is kept so low, we're pushed in the equilibrium this way, as opposed to GAP going back to DHAP. All right, so let's review stage one. We finished stage one, and in stage one molecule of glucose has been transformed to 2 molecules of glyceroldehyde 3-phosphate, and we have consumed 2 molecules of ATP in order to do that conversion. So now we're going to continue on to the beginnings of stage 2. All right, so now we are about to create ATP. So in this one, we're going to have oxidation of G3P. So glyceraldehyde 3-phosphate is oxidized using NAD+, and then phosphorylated in order to make 1,3-bisphosphoglycerate, or 1,3-BPG. This is going to produce, also as a side product, NADH, or the reduced form, as well as an acidic proton. So again, G3P undergoes oxidation and phosphorylation in an oxidation reduction reaction governed by glyceraldehyde 3-phosphate dehydrogenase to form 1,3-BPG. This enzyme is actually a tetramer of 4 identical subunits, and each subunit can bind G3P and NAD to catalyze the reaction. So we can see how we can quickly consume GAP, keeping those concentrations low in order to produce 1,3-bisphosphate glycerate. This reaction is exergonic due to the oxidation of the aldehyde group by NAD+. So that releases enough energy, just enough energy to drive the phosphorylation at the C1 of glyceraldehyde 3-phosphate, as we see here. In step 7, we have a phosphoryl group transfer. So we're going to produce our first molecule of ATP. So in this step, ATP is synthesized as phosphoglycerate kinase. This guy catalyzes the transfer of the high-energy phosphoryl group of 1,3-BPG, which is this group on here on C1, to ADP. So this is a great example of substrate-level phosphorylation. This is different than oxidative phosphorylation because we're transferring a phosphate group from one molecule to another, as opposed to doing oxidation reduction reactions of inorganic phosphate coupled to a molecule. Because 2 molecules of 1,3-BPG are produced per molecule of glucose, we get a total of 2 molecules of ATP per molecule of glucose in this step. Breaking the C1 phosphoryl bond releases approximately 50 kilojoules per mole, where it takes 30 kilojoules per mole to make ADP into ATP. So again, we're releasing 50. We need 30 to make ATP, so we have a net of 20 kilojoules per mole overall in this reaction, making it very thermodynamically favorable. Step 8, we have interconversion of 3PG, which is our product after we remove that first phosphate group from C1, to 2-phosphoglycerate. And the reason we do this is because this sets us up in the scene for the next reaction, as you'll see. So cells convert 3PG to 2PG, so it's just a isomerase reaction, through a 2-step addition-elimination reaction. This step in glycolysis sets us up to make phosphoenolpyruvate, or PEP, which has a much higher energy release upon dephosphorylation than 3PG. For instance, 3PG only releases 12.6 kilojoules per mole, where PEP releases 61.9 kilojoules per mole of energy. So the first step in making PEP is to transfer from carbon 3-phosphate to the carbon 2-phosphate. Okay, so remember 2,3-BPG that binds hemoglobin. So 2,3-PPG is actually a byproduct of glycolysis. So when we have glyceraldehyde 3-phosphate gets phosphorylated into 1,3-bisphosphate. 1,3-bisphosphate, if it accumulates in high amounts, can be used by another enzyme, bisphosphosylserate mutase, to make 2,3-BPG. 2,3-BPG can also then be dephosphorylated and made into 3-phosphoglycerate. However, we will not get our ATP molecule if we take this route. All right. So what's interesting is if we look at our oxidation saturation graphs that, remember, we were looking back at when we were talking about hemoglobin, the red line is our normal erythrocytes. Okay, so this is your oxidation saturation based on the partial pressure of oxygen of hemoglobin. If we have hexokinase deficient cells, okay, so what this means is we cannot convert glucose to glucose 6-phosphate, okay, which is that first sort of committed step in glycolysis. What that means is eventually we won't ever get to produce 2,3-BPG because that is a byproduct of glycolysis. So if we never undergo glycolysis, we'll have lower amounts of 2,3-BPG, which means we will inhibit, or we will not inhibit, oxygen binding. Because remember, 2,3-BPG binds to the T state of deoxyhemoglobin, preventing oxygen binding. So if we decrease 2,3-BPG, we will increase oxygen binding. However, if we have pyruvate kinase deficiencies, so pyruvate kinase is further down in the, isn't this pyruvate kinase? No, that's phosphoglycerate kinase. Okay, pyruvate kinase is further down in glycolysis. It's one of the last steps of glycolysis. So if that's deficient, we'll actually get a buildup of metabolites occurring. So remember our red light. If we have a red light on our street, all the metabolites behind the red light, or all the cars behind that red light, will start to build up. So if pyruvate kinase is a red light that's further down in the metabolic pathway of glycolysis, we'll get a buildup of all of these metabolites, including eventually 2,3-BPG. So if we get a lot of 2,3-BPG, we'll see we will decrease our oxidation saturation. So this is just a pathway in which cells can regulate 2,3-BPG, and they regulate it through the regulation of glycolysis. All right, so now coming back to glycolysis, step 9. So let's go back to step 8 real quick so we can remember where we are. So in step 8, we have 3-PG, and we did a isomerase or a mutase reaction to make 2-PG. So remember, this is setting us up to make PEP. So in step 9, we have dehydration of 2-PG. So we remove a molecule of water using the enzyme inalase in order to create now phosphoenolpyruvate, or PEP. So just to recap, inalase catalyzes the dehydration of 2-PG to form PEP. Now remember, PEP is a very high phosphoryl group transfer potential, much higher than 2-PG or 3-PG, because it contains an enol phosphate group here instead of just a simple phosphate ester here. Okay? So the energy released upon the hydrolysis of the phosphate in PEP now is negative, so we get a total of negative 60 kilojoules per mole of energy released when we cleave this bond. If we had cleaved it here at this stage, we would only probably get about, I think it was around 12. So 12 versus 60. Now, step 10, so our final step of glycolysis, is the synthesis of pyruvate from PEP. So in the final step of glycolysis, pyruvate kinase, right here, catalyzes the transfer of a phosphoryl group from PEP. So from PEP, this phosphate group gets transferred to ADP to make ATP. Again, remember, because we have 2 molecules of pyruvate for every 1 molecule of glucose, we are then going to get 2 molecules of ATP per 1 molecule of glucose. PEP is irreversibly converted to pyruvate because in this reaction, the transfer of the phosphoryl group from a molecule with a higher transfer potential to one with a lower transfer potential occurs with a large free energy loss. So remember, to make ATP, we need 30 kilojoules per mole. When we cleave this bond here, we're going to release around 60 kilojoules per mole, giving us a net of negative 30 kilojoules per mole for every phosphophenols we convert to pyruvate. And remember, so this is the pyruvate kinase. This is the enzyme I was talking to you about here in this table. So if pyruvate kinase is deficient or inhibited, we will end up getting a buildup of metabolites upstream, including a buildup of 1,3-bisphosphate glycerate, which can then be converted into 2,3-bisphosphate glycerate. So you can see how that relationship, by inhibiting pyruvate kinase, we can increase 2,3-bisphosphate glycerate concentrations, which will then decrease hemoglobin's affinity to bind oxygen. So let's review stage 2. So in stage 2, remember, 2 molecules of pyruvate and 4 molecules of ATP are formed, and 2 molecules of NAD+ that oxidizing agent are consumed for every 1 molecule of glucose that undergoes glycolysis. So overall, between stage 1 and stage 2, we net 2 molecules of ATP total for every molecule of glucose. So let's recap. Remember our fate of pyruvate. So pyruvate is also a product of glycolysis, as you can see here. Pyruvate is still an energy-rich molecule which can produce substantial amounts of ATP, and so it can be shuttled through for aerobic respiration through the citric acid cycle, or it can go through 2 routes of anaerobic respiration. If it's in like eukaryotic cells, it's undergoing homolactic fermentation. If it's prokaryotic cells, we are undergoing alcoholic fermentation. So again, this is prokaryotic and eukaryotic, anaerobic. So these are 2 graphs of the free energy changes during glycolysis. And I just wanted to take a minute to look at this. So this first line up here, this is our delta G naught prime. Okay, so this is under standard conditions of 1 atmosphere, 25 degrees C, and at 1 molar concentration. So if you notice, there's really not a clear pattern in here. We do see an overall negative delta G. However, but if we look at under the conditions of the cell, so this is delta G in the cell of glycolysis, we can see a pretty clear pattern. So here's that step 1, okay, where we go from glucose to glucose 6-phosphate. Then we have step 2. Then we have step 3, which is another very large negative delta G. So this is where we do the second phosphorylation step. Then we have a bunch of steps that are near 0 or near equilibrium. And now we have step 10, I think. Yeah, because this is lactate here. Okay. So we have step 10, step 3, and step 1. And you can see all of those have very large negative delta G's, where the other ones stay pretty level. Your book kind of graphs it this way, and it's a little harder to see, but you can still see 1 is very negative, 3 is very negative, as well as 10, where the others stay pretty near equilibrium, or pretty much near a delta G of 0. Okay, so this is going to be important when we talk about the regulation of glycolysis. So it's not surprising if you look here at steps 1, steps 3, and steps 10 that all of the enzymes that regulate these large negative delta Gs are our key enzymes for regulating all of glycolysis. So hexokinase governs step 1, PFK1 governs step 3, and the pyruvate kinase governs step 10 in glycolysis. Now, hexokinase, remember I said, can be inhibited by glucose 6-phosphate, its product, as well as ATP. PFK1 can be inhibited by citrate as well as high levels of ATP, citrate being a byproduct of the citric acid cycle. And then pyruvate kinase can also be inhibited by a product of the citric acid cycle, acetyl-CoA, as well as ATP, which, ATP is our end product in all of these cycles. Activators of some of these enzymes include fructose 2,6-bisphosphate, as well as AMP for PFK1, and we'll talk about this in a little more detail in just a second. And then pyruvate kinase is activated by fructose 1,6-bisphosphate. So if you remember, fructose 1,6-phosphate is a product higher up in glycolysis. So this is an example of feed-forward activation. And both of these are activated by AMP, which makes sense. If the cell has a lot of AMP, then obviously it's ATP deficient, and so glycolysis will be favored. Now, glycolysis can be also regulated by hormones. So two such hormones are glucagon and insulin. So glucagon is a hormone released by pancreatic cancer cells when blood glucose is low. It activates an enzyme called PFK2, which is like PFK1, but it's PFK2, which reduces the levels of fructose 2,6-bisphosphate in the cell. So remember, fructose 2,6-phosphate is an activator of PFK1. So if we reduce these levels, then we're going to reduce the activity of PFK1, which is going to reduce the flux of glycolysis, because it's going to slow glycolysis down. Insulin is also a hormone released from pancreatic cells, but it's released when blood glucose is high. It activates PFK2, which then increases the level of fructose 2,6-bisphosphate, remember this guy, in the cell, which then will increase activity of PFK1, which will then increase the flux of glycolysis. So it increases the breakdown of glucose. Okay, so that's all I wanted to go through with you guys today. Make sure you can go back to your learning objectives. Make sure you understand what it is that I'm asking you to be able to describe and identify and explain in all of these questions. And I will see you in class on Wednesday. Hope you have a great day. All right, now, trying to remember where I go to. Oh, this guy.